Hello, my name is Amber Jump, and I'm in the North Idaho College Medical Laboratory Technology program. This is my capstone project, how to perform a gram stain and how to read it. So what is the gram stain? What is it used for? A gram stain is a test used to help identify bacteria. It's usually one of the first beginning types of tests for microbiology. Of infected tissues and secretions in patients. So, what specimens are acceptable for this test? Well, some of the ways providers can collect gram stain test samples include brushing or scraping of tissue, collecting fluid or discharge samples, and fine needle aspirations to draw a fluid from a sample. What specimens are acceptable for this test? Part 2. The sites that are collected by a swab can include the throat, the nostrils, genitals, and the skin wounds. Samples that you can collect with a sterile container is spit or phlegm. It's usually called sputum, urine, pee, and then stool, which is poop. Areas in your body that may require the fine needle aspiration include places that usually shouldn't be exposed to, like air and stuff, like the fluid in your joints, the fluid around your heart, the fluid around your lungs, and cerebral spinal fluid, which is around your spinal cord. All of these are put into sterile containers and then sent to the laboratory. So the last part, the part that we get to do in the lab, is all of these previous samples need to be on these type of plates. For other labs, it could be different, but most labs that I've seen on internship will just do these basic 4 just to cover all the basis of bacteria. So a BAP is blood agar. It's 5% sheep blood, and it doesn't have any inhibitors. It will grow anything. Well, if they want to grow, they'll grow pretty much. MAC or McConkey agar, it's gram-negative oriented. And chocolate agar has human and XMV factor for fastidious organisms. CNA or Columbia nalidixic acid agar, which usually also has 5% sheep blood. That's usually the opposite of a MAC plate, which produces gram-positive organism growth. Transport and storage conditions for the specimens. Transport usually is at room temperature unless otherwise specified. Delayed transport depends on the type of specimen, but usually refrigeration and putting it on ice is the typical delayed procedure. Specimen processing required. Some of the commonly used specimens to process in the lab are sputum, blood, cerebral spinal fluid, acetic fluid, synovial fluid, pleural fluid, and more. They all need to be set up in a specific way, but each lab can be different, so you would have to read their own protocol in each order because there might be different orders. Swabs from the nostrils, throat, rectum, wounds, and cervix, etc. need to be set up in a certain way also. So getting back into the gram staining, this is the pre-staining procedure. All of the things that we need to do to the slide before we actually go in with the gram staining kit. So we will put a pencil to label the slide and not pen because we have a procedure coming up in the staining procedure that will completely obliterate the pen and we won't be able to tell what we wrote because it will be washed off essentially. So pencil is the way to make it stick. After we label the slide, we're going to put 1 drop of water for each patient slide that we're going to work with. Then we're going to grab the plate with either the patient or QC on it. So we can either grab it with a loop or different types of loops like wire loops or plastic disposable loops. And there's also loops with different sizes. So usually we'll just go with 1 small isolated colony, or even that might be too much for a gram stain. So for staph, usually I've been able to get a pretty good gram stain with just 1/4 of a small colony. So it depends on the size of colony and the size of loop, I guess, that you're using. Gently mix the colony into the drop of water that you put on the slide. This is really important because you don't want to mix too hard, or you could change the arrangement when you look at it later, like staph versus strep. Once mixed, put the slide on a slide warmer to heat fix the bacteria to the slide so we don't have everything washed off and not see anything once we put it under the microscope. The slide warmer should be about 56 degrees Celsius or 132.8 degrees Fahrenheit. For the MLT program, heat is the primary fixing method. I don't think we've ever used methanol to fix bacteria to the slide, but I have seen videos and people at my internships do this method. So usually if you want to methanol fix it, you'd leave the slide on a paper towel or absorbent pad and pipette the methanol to flood the slide, and then you would leave it on the slide for 2 minutes. How to perform the test. The main question that we're going to ask on this is, are we trying to answer, did grams iodine retain in the cell? It's either a yes or a no answer based on the result, the final gram stain, basically. So I'll get more into that in the next couple of slides. So finally, we have the staining procedure. We have the step 1 through step 9. Crystal violet, and all of these stains I'll show in my demonstration video, I believe, right after this slide. But we do crystal violet for a minute, rinse with DI water, make sure it runs clear, and you'll see that in my video. When it runs clear, then you stop. Then we do grams iodine for 1 minute also, rinse again, and then here's a really critical, like I would say the most critical step of gram staining, and it could mean, what I mean by critical is like you probably will have to redo it if you don't do it right. So we put the decolorizer on for about 1 to 4 seconds. That's a huge difference between a minute and 30 seconds and 1 to 4 seconds. So pretty quick. All of the kits at NIC that I've experimented with are very fast decolorizers, and that's because they have 3 ingredients in them, and I will show that in the demonstration video. Rinse immediately with DI water. We really don't want the decolorizer on for more than 1 to 4 seconds long, or otherwise that could cause false results. Then the last 2 steps, we do safranin, the counter stain, for 1 more minute, and then we rinse that until it's completely clear, and then drain it and air dry, or we could blot with bibulous paper, which I do demonstrate in the other video. So here's the video. So we have crystal violet, a grams iodine, a decolorizer, and saffron and stain. So I'm going to show you, swirling it around, that it's pretty purple, pretty violet. Makes sense. Iodine's supposed to be yellowish, like all food color dyes. Yellow kind of looks red, but when you swirl it around, it kind of turns yellow. The decolorizer is clear. And the 3 ingredients are ethanol, methanol, and acetone, so it's pretty potent. And then there's saffronin, which is supposed to produce a pink, reddish type dye. So I'm going to be working with E. coli and Staph aureus. Those are the dates that were the bacteria were plated. So we got an older one and a younger one. Staph is the more younger colonies look less spready. The E. coli is older, so it's more spready. So we have sandblasted slides to work with. I'm going to grab 2, one for each organism here. So we got to label the slide with 2 patient identifiers, or just 2 identifiers is good habit for QC as well, to truly know what you're working with. So I wrote down staph and the MRN, or not MRN, but accession number. Then we have E. coli, and then the accession number that goes with that. So we add a drop of water to each slide. And I am using DI water, deionized water. So I'm using a smaller loop, trying to focus for you. It's way smaller than the other ones. Then we're going to pick our individual isolated colonies for each slide. We got E. coli first. So it's a little spready. So I'm trying to pick just a little bit from one colony. And then stirring it faster than this will actually cause it could cause breaking the arrangement of cells if you stir and mix with the water faster than this speed. So you got to be careful with how fast you're mixing organisms too. So now we're going to do the staph, and we're going to do the same thing. I'm going to try to show you how I pick my colony. So that one looks pretty isolated, so I'm just going to take a little bit. It's actually kind of a lot because staph goes a long way. So mixing it, spreading it out because we don't want it too thick, or otherwise we can't see very well because it picks up too much of the stain. So now we're going to heat fix it. We have a slide warmer. So I'm going to put both my slides on the slide warmer, and once it's done, we're going to start our staining process. So I apologize that I didn't film this. I thought I originally did, but I am already on the iodine step. I did do the crystal violet step, but for some reason, my camera didn't film it. So it's been a minute. I'm speeding it up for video purposes, but I'm rinsing off my iodine, and you can see it's kind of purple. So I did do crystal violet before this. So I did crystal violet, and then I did rinse, and now I'm doing the iodine and rinsing. We're doing the decolorizer, and we're going to do it until it runs clear. All of the purple is now on the edge of the slide, so that's clear to me. So we're going to rinse that off. Notice how the bacteria doesn't look purple anymore as much as it did right after the iodine step. And then after the decolorizer, we're going to do safranin, our counter stain. I'm going to leave that on for a minute. And then rinse it off with DI water. So our next step is to blot, get that excess water and excess droplets of stain off onto some clean paper towel. This will make it dry faster, air dry faster. Or we could use Biblia's paper. It's a little booklet where you gently press down and it will soak up all of the excess water, but pressing it in that motion won't take the bacteria off. So I was going to show how to read it. So this is the part where we're going to read the gram stain. So I have my microscope. I put my slide on my microscope, and we're just going to jump straight into the oil immersion setting. So the ocular lens that I'm going to look through first is a 50x. Scanning, trying to get a good feel, looking at the gram stain, seeing how well the gram stain is in regards to is it all purple or is it all pink? Is it a mixture of the two in some places? Is your stain, is the organism too thick? Are you able to actually see what the organism is because there's too much or too little of it? So the 50x will give you a good judgment of that type of question. So I'm going to pause right here because this is what I was originally going to show you. This is how the 50x looks, and I found a pretty good spot where I could read and I could really make sure that this is a gram-positive staph. They are little coxy in clusters, perfect as they should be. And we would also expect this organism to be gram-positive, so that's good. And then I'm going to skip this to my 100x. So from the 50x ocular lens, we went to the 100x ocular lens. And as you can see, this is so much clearer, more definitive. Little grape clusters, they look perfect like that. And you can really tell that they are obvious clusters and not strep-like. So I didn't stir too hard, like I said in that one step, mixing your organism with your DI water before in the pre-staining procedure. It's very important. Then we're going to skip a little bit. And then I'm taking that off because I think it was good. And now we're going to put the E. coli on. And this is 50x. I've zoomed it in a little bit because my picture wasn't quite clear, but this is 50x ocular lens. And as you can see little bacilluses in this really nice thinned out area. They're kind of pink, so that's good. I don't really see any purple. And we would expect E. coli to be quite pink. So here it is on the 100x, and also zoomed in since my picture is a little blurry, and I apologize for that. But we can see almost a reddish pink, like I was explaining safranin is notorious to be. So in my opinion, this was a very good gram stain of E. coli. Gram negative is gram negative for a reason. There is no coincidental like reaction unless if you over decolorize, which would be the only case something would be gram negative falsely or gram-positive falsely would be that decolorizer step. So gram-positive bacteria have significantly larger peptidoglycan layers on their cell wall, on their outer membrane, essentially. So they soak up that crystal violet a lot more than the gram-negative can. So I'm not saying that gram-negative organisms can't, but they tend to pick up safranin way more easily because of the time step. So gram-positive, it'll take much longer for that decolorizer step to really get all of that crystal violet out because there's so many layers to get through. So it's not coincidental, it's science, so that's super cool. So, like I just showed in the demonstration video, the microscopic examination should be used at the 40x to evaluate the smear. I use 50x. The microscope I was using didn't really have a clear field of view when I was on the 40x, so I thought it would be a little bit better to show you guys through the demonstration of a 50x, even though it was an oil immersion lens as well as the 100x. So I did use 50x, but 40x and 50x are usually interchangeable. So 50 and 100 is what I use in the video. All areas of the slide require an initial examination. Areas that are only 1 cell thick should be examined, and also the thick areas in the slides often give variable incorrect results because you can't really see what's going on. They're too thick, so that kind of makes sense, right? With direct gram stains, or depends on where your sample is coming from, the white blood cells and the macrophages will stain gram-negative, and squamous epithelial cells will stain gram-positive. So how to read the results through the microscopic examination. So based on how to read this, the morphology and arrangements will really help you with what you're looking for. So bacteria has morphology, and then they have arrangements. So as you can see in the picture on the right, they're organized into cocci, bacilli, and spirochetes. So we can see that there are staphylococci, there are streptococci, and diplococci, and tetrads. So staphlio, or staph, is usually clusters. Strepto, or strep, is chains, and diplo is pairs, usually pairs of 2. So, and that can be interchangeable with bacilli. There's diplobacilli, streptobacilli. So it's just the arrangement of how these bacteria are. So, and spirochetes are their own thing. They're just little silly noodles. So, interesting, but definitely will help you in describing your gram stain and seeing, truly capturing what you're seeing there. So staph is usually a common gram-positive organism, and bacilluses are usually the common gram-negative organism, which is why in my demonstration video I used E. coli and I used Staph aureus. So they were prime examples for a gram stain. So relating on that topic, gram-positive cocci, they retain the crystal violet and safranin. So those thick peptidoglycan layers I showed earlier, they actually retain both, making it super dark purple. And these aren't my pictures either. I grabbed these from the internet, but I thought they were great illustrations of what the interpretation of gram-positive cocci are. So some of the examples of gram-positive organisms are for cocci and bacilli. So I'll start with cocci first. Staphylococcus species and Streptococcus species, which can be quite a bit, as you'll see in the NIC microbiology courses, and Bacilli, Corneobacterium species, Clostridium species, and Listeria species, which the program doesn't touch up too much about, more so on the Enterobacteriaceae family, so pay attention to those guys as well. I'm going to take my word back on that because Enterobacteriaceae is gram-negative. I apologize. Gram-negative rods, they're not usually gram-negative cocci, so you should expect the gram-negative cocci to be an over decolorization usually, but there are always exceptions to biology, and there are real gram-negative cocci, but they're usually not as common. They don't retain crystal violet stain, and they only soak in the safranin based on the time it takes to decolorize. So it will appear pink to red, and I also grabbed this image from the internet. So good representation. The examples for cocci are Neisseria gonorrhea, Neisseria meningitidis, and Marxella species. The bacilli for gram-negative organisms are Escherichia coli, E. coli, Pseudomonas species, Proteus species, and Klebsiella species, all a part of the Enterobacteriaceae family, as I was saying earlier. How to report the results of gram stain. So the bare minimum of a recorded result should contain the following. For the morphology, either cocci or bacillus. Arrangement, singles can be quite different depending on what sample you're working with, but usually chains and clusters are the most common. But single cell, diplococci, tetrads, curved, palisades, and spirals also occur. And of course, the gram reaction is either gram positive or gram negative. So that's how you report bare minimum gram results. Pre-analytical errors which might affect the test are poor specimen quality, or the bacterial culture was too old. I'm not too sure about bacterial culture being old because I've never actually done it myself, but I had seen this come up a lot, so I just put this here because it might be a factor. I think it might have to do with gram results becoming gram variable and not as strongly associated with either gram positive or gram negative because the cell membrane breaks down over time. So I think that might be the reason why it would be bad to do an older bacterial culture. Usually 18 to 24 hours is optimal for gram stain, or at least a culture growth time anyways for gram stain. So there's the pre-analytical errors. Many analytical errors that could affect the test would be the slide being inadequately or over decolorized with the acetone/alcohol mixture, and this results in a false reading or having to redo the test. Most likely having to redo the test if you catch yourself not decolorizing it quickly, or not catching yourself. So the next one is when using a wire loop after reincineration. It was still too hot when touching the bacteria, so you basically killed it, and it doesn't have any morphology or arrangement whatsoever. It's just dead. It's just a glob together, I guess, because you flame your wire loop to rid it of any pests when you want something to be sterile. So that would be that analytical error. And failing to perform any of the 4 steps plus water rinses, if you don't follow those 9 steps that I provided earlier, it can result in an imperfect gram stain, which might cause false results as well. Smear prepared too thick, can't see through it, can't read what it is, you're going to have a hard time. You're probably going to have to redo the test. So, too thick of a smear is a bad thing. And excessive heat fixing, that can also almost like the wire loop analytical error, where you would essentially burn the bacteria to the point where it can't actually be properly stained as well. So post-analytical errors, which might affect the test, only 2 things I could find really, which would be reporting an incorrectly read gram stain that could change the patient outcome. It might be worse, might make them worse than what they were. So I really think you should, anyone going into this field should double check the gram stain reportings to make sure that it makes sense with what you're seeing on the plate. Because your patient depends on it, really. Sometimes, most of the time, when they're in, ordering it stat or something like that. And reason being, gram stains can be read wrong. So, and thank you for my capstone project. And here are the references I used. Thank you.